So we shall understand this concept of bacterial density determination with this second example. So here the example goes like this. In a bacteriological test conducted on a sample of water, the following results were obtained. 3 positive in 5 10 ml portions, 1 positive in 5 0.1 ml portions, and 2 positive in 5 1 ml portions. So given this data, we are asked to calculate the most probable number of coliform organisms and comment on the result. So according to Thomas, MPN is equal to 100 into number of positive portions in the denominator, ml in all negative portions and ml of all portions. So we have to now first of all calculate the number of positive portions. So as it is given in the data, that is in the problem, 3 positive in 5 10 ml portions. Therefore 3 plus in the second set, say where 1 ml sample was poured, there 2 positive in 5 1 ml portions, therefore it is plus 2. And in the third set where 0.1 ml sample was taken, only 1 was positive, therefore plus 1. So total number of positive portions now will become 3 plus 2 plus 1, it is equal to 6. It means that 3 sets of 5 test tubes, therefore 3 into 5, 15 test tubes were taken, in which already nutrient medium was there, to which we have poured the water sample quantity of 10 ml to the first set and 1 ml to the second set of 5 test tubes and 0.1 ml for the third set of 5 test tubes. And now when all these 15 test tubes are kept in the incubator and after say 24 hours where temperature was maintained at 35 or 37 degrees centigrade, if any bacteria are there, they are going to ferment the content and therefore the result of which will be the production of the gas. So out of these 5 test tubes, 3 plus 2 plus 1, so totally 6 test tubes gas was formed. Therefore number of positive portions are 6. Then in the denominator we have the first term ml in all negative portions, means the total quantity of the sample taken to the test tubes where there was no gas formation. So here in the first set, out of 5 test tubes, 3 indicated positive. Obviously 2 test tubes indicated negative. Therefore quantity of the sample taken in the negative test tubes will become 2 into 10, that is equal to 20 ml. And in the second set of 5 test tubes where 2 were positive, obviously 3 test tubes indicated negative where 1 ml sample was taken, therefore it is 3 into 1, therefore it is 3 ml. And in the third set of 5 test tubes where 0.1 ml of the sample, water sample was dispensed, only 1 indicated positive, obviously the remaining 4 indicated negative. Therefore quantity of the sample in the negative test tubes of the third set will become 4 into 0.1, therefore it is 0.4 ml. So if you add all these quantities, say 20 ml plus 3 ml plus 0.4 ml, so ml total ml in all negative portions will now become 23.40 ml. Now the last term in the denominator is ml of all portions, means we have 10 ml portions, 1 ml portions and 0.1 ml portions. What is the total quantity of the sample taken, that is in terms of ml of all portions will be, say 5 test tubes, 5 into 10 it is 50 ml, 5 into 1 ml, that is second set, that is 5 ml, and in the third set we have dispensed only 0.1 ml, therefore 5 into 0.1 it is 0.5 ml. If you add all these, total quantity of the sample will be 55.5 ml. So after calculating these required parameters, then if you substitute in the given standard formula as per the Thomas, it is 100 into number of positive portions will become 6, and in the denominator ml in all negative tubes it is 23.4 ml, and ml in all portions it is 55.5 ml. If you simplify this, we are going to get most probable number as 16.65. 16.65 number of bacteria are there. So the value should be 0. Therefore, the MPN index of coliform bacteria should be 0 or less than 1. However, therefore this particular water sample or the source, before it is supplied, it should be disinfected properly with appropriate methods of disinfection. Then only the water will become safe to drink and other consumption, say for the food preparation, etc. And also we shall take now the another example, second or the next example. The following are the results of a bacteriological test on a water sample. Determine the MPN and state whether water sample tested is suitable for drinking or not. See, the data has been given here in the form of these tables. Number of test tubes tested are 5 in the first case, where sample concentration was 10 ml, and number of positive test tubes are 4, and number of negative test tubes are say 1. Means in the first set of 5 test tubes where 10 ml water sample was dispensed after incubation period, that is when these test tubes are kept in the incubator at say 37 degrees centigrade, say for 24 hours. When they were taken out and observed, the gas was formed in the 4 test tubes where 10 ml portion was there. And in the second set where 1 ml of sample was dispensed, 2 test tubes indicated positive result, and obviously 3 test tubes indicated negative. And in the third set where 0.1 ml sample was taken, only 1 test tube indicated positive, obviously remaining 4 tubes indicated negative, no gas formation. Lastly, where 0.01 ml using a micro pipette the sample was dispensed, then it was observed that 0 test tubes, means all the 5 test tubes indicated negative only. So with this data we are asked to calculate the MPN value. So in the Thomas equation we need to calculate the number of positive portions, ml in all negative portions, and ml of all portions of the sample. So firstly, ml of all portions, we have in the first case 5 test tubes where 10 ml was poured, and then second set 5 into 1 ml, and third set of 5 test tubes 0.1 ml, therefore 5 into 0.1 ml, and in the third set where 0.1 ml of the sample was poured, and in the last set 0.01 ml was poured. So here number of positive portions will be, in the first test 4 indicated positive, second set 2 indicated positive, third set only 1, fourth 0. If you add all these, number of test tubes giving positive results will become 4 plus 2 plus 1, it is equal to 7. Now ml of all negative portions indicated, in the first set when 4 are positive, 1 indicated negative, where quantity of sample taken was 10 ml, therefore 1 into 10, plus in the second set where 2 portions indicated positive, 3 indicated negative, therefore 3 into 1 ml sample was taken, therefore it is going to be 3 ml. And in the third set where 0.1 ml sample was dispensed, only one was indicated positive, therefore ml in negative portions will become 4 into 0.1 ml, that is 0.4 ml. Lastly, in the last test tube where 0.01 ml was dispensed, number of positive portions were 0, but therefore all the 5 test tubes indicated negative, therefore quantity of the water sample taken in the negative test tube here will become 5 into 0.05. So if you simplify this, now ml of all portions will become 55.55 ml, ml of all negative portions will become 13.65 ml, number of positive portions 4 plus 2 plus 1 it is equal to 7. When you substitute all these three values in this standard formula given by Thomas, 100 into number of positive portions divided by under the root ml in all negative portions multiplied by ml of all portions, here MPN is equal to 100 into 7, number of positive portions here 7, then in the denominator under the square root ml in all negative portions is 13.65 and ml of all portions it is equal to 55.55. If you simplify these numbers equation, you are going to get 25.42. Here also, since the MPN is more than unity, it indicates the water contamination by the coliforms. Therefore, it requires the removal of this by disinfection. So, this is about the determination of the most probable number. So, one more example for your better clearance or better understanding of the doubts. The following are the results from a bacteriological analysis of a water sample. Calculate the MPN coliforms. See, sample size, portions and positive samples. In the previous example they gave also the negative samples also. Here only positive samples are given. Obviously we can determine the negative samples also. So when the sample size or concentration was 10 ml, number of portions, such portions of 10 ml taken were 5 test tubes and among them all the 5 indicated positive. Therefore obviously negative samples will become 0 in the first case. When sample size was 1 ml, portions are taken say 5, so then the positive portions in the second case it was 4, obviously 1 test tube indicated negative. In the third set where 0.1 ml was the sample size, number of portions are again 5 here, only 2 portions indicated positive, therefore obviously 3 are negative. Lastly, in the last set where 0.01 ml was dispensed for all the 5 test tubes, only 1 indicated positive, obviously remaining 4 indicated negative. Now we shall calculate the required parameters. So first of all, let us calculate the number of positive portions. See, number of positive portions are 5 plus 4 plus 2 plus 1. 5 plus 4, 9, 9 plus 2, 11, 11 plus 1. It is totally 12 numbers. Number of positive portions are 5 plus 4 plus 2 plus 1. It is 12. Then ml in all negative portions. Here you see number of negative portions. In the first case, when out of 5, all 5 samples indicated positive, there were 0 samples indicated negative. Therefore 0 into 10 ml sample was taken, therefore this quantity will become 0. Plus in the second case where 4 indicated positive, 1 indicated negative, therefore it will be 1 into where 1 ml portion was taken, it is therefore 1 into 1, it is equal to 1 ml. Then in the third set where 0.1 ml was dispensed, 2 indicated positive, obviously 3 will become negative, therefore it is 3 into 0.1, therefore the value will become 0.3. Now in the last set where 0.01 ml was the sample size and only 1 test tube indicated positive, obviously 4 are negative, therefore 4 into 0.01 therefore its value will become 0.04. If you add all these quantities, therefore this 1 into 1 is 1 plus 3 into 0.1 is 0.3, then 4 into 0.01 it will become 0.04. If you add all these things, it is 1 plus 0.3 plus 0.04, it is equal to 1.34 ml. And calculating the ml of all portions, we have 5 test tubes of 10 ml, second set 5 test tubes where 1 ml sample was put, and in the third set 5 test tubes of where 0.1 ml sample was taken, and in the last 5 test tubes where 0.01 ml was taken. Therefore total quantity, that is ml of all portions taken in the test tubes in the entire test, will become 5 into 10 50 plus 5 into 1 5, that is 55, plus 5 into 0.1 0.5, therefore it is 55.5, and also lastly 5 numbers into 0.05, value will become 0.05. If you add all these things, we are going to get 55.55 ml. If you substitute these quantities like 12 positive portions, ml in all negative 1.34 ml, and ml of all portions 55.5, we are going to get the result as 13.91. 13.91 is the MPN value. Therefore it is not asked in the example to comment on the result. However, we can comment that as this MPN value is more than 1, so therefore it requires disinfection, that is to destroy or kill the disease causing bacteria, and in order to make the water safe for drinking and other oral consumption purpose. So this is about the determination of the most probable number of coliforms, and also we shall now discuss about the waterborne diseases.